Hello everyone, and welcome back to AP Bio with AP Playground. Today, we're covering topic 5.4, Non-Mendelian genetics. So in 5.3, we discussed Mendelian genetics and patterns of inheritance. However, not all inheritance of traits follow these patterns. And so when we look at things like linked genes, codominance, incomplete dominance, sex-linked traits, or even non-nuclear inheritance, so not even from DNA in the nucleus, then we have to look at some different patterns of inheritance. And we'll be discussing those non-Mendelian genetics in this video. And our objectives are suggested skills, statistical tests, and data analysis 5A, perform mathematical calculations, including mathematical equations in the curriculum, means, rates, ratios, percentages, and percent changes. And we'll be looking at that when discussing linked genes. And then 5C, perform chi-square hypothesis testing, testing whether the observed is different from the expected amount. So when discussing dominance before, such as in 5.3, we've been talking about complete dominance, where the dominant allele is always going to take control. So no matter whether you have a homozygous dominant, so let's say our allele is P, so if you have two capital P's, or a heterozygous genotype, so one capital, one lowercase p, then the dominant allele, so this capital P, would take control in both situations, and the phenotype exhibited would be of the dominant allele. So if P stood for purple, then and these were flowers, then both of these flowers would be purple, even though the heterozygous flower would have a recessive allele. However, this is not the only kind of dominance. There's also incomplete dominance, where heterozygous organisms are going to have a phenotypic mix between the two alleles. So for instance here, where we have a homozygous dominant and a homozygous recessive plant for red and white roses, the offspring is going to end up being heterozygous but pink. So the alleles have mixed, blended together to create this incomplete dominance. There's also codominance, where we have this homozygous dominant red flower and homozygous recessive white flower. The alleles are expressing themselves in a heterozygous flower where some petals are red and some petals are white. So both alleles are dominant, so there's codominance, two dominant alleles, and they both affect the phenotype in different but equal ways. One does not have control over the other, like in complete dominance. There's also the situation of multiple alleles, and that's just like what it sounds. These are genes which exist in more than two allele forms. There are multiple alleles for that gene, and the most common example of this, one of the most, would be human blood types. And there exist three alleles for human blood types. There's IA, IB, and lowercase I. So these two are both dominant, and so when you have a blood type that is uppercase IA, uppercase IB, then you're experiencing codominance, and that's creating type AB blood. When you have an IA and a lowercase I, then you have regular complete dominance where the capital IA is taking over, and so you have A-type blood. Same thing with capital IB, that's going to result in B-type blood. And then when you have two of the recessive lowercase I alleles, then you're going to have O-type blood. So within these multiple alleles, we can see these other forms of dominance that we discussed, including complete dominance, but also codominance. Now to talk about linked genes. So genes are genetic information, and as such, they have specific locations on chromosomes, because chromosomes and DNA is what's storing that genetic information. And linked genes are on the same chromosome and tend to be inherited together during cell division. And the only way they could be separated is due to crossing over. So if we look down here, when we have our homologous chromosomes that are getting ready for meiosis, if no crossing over occurs, then all of the four haploid daughter cells that come out of meiosis are going to have a chromosome that is identical to what the parents had. They're going to be parental type. That will be their genotype and their phenotype. However, if crossover does occur, then we have a swapping of genetic information, creating daughter cells that have non-identical genetic information to the parents, and these are called recombinants. And a linkage map can show how close genes are on a chromosome. Now, genes that are further apart from each other have a higher chance of crossing over and causing recombination. Because, for instance, if gene B was actually located up here right next to gene A, then there is a very low chance that crossing over would occur all the way here right in between gene A and gene B, especially because crossing over generally only takes the ends of the chromosome and swaps them. And so there's a lesser chance of them being separated. And so linked genes that are closer together are generally going to stay linked through meiosis. However, genes that are further apart have a higher chance to be separated. So now to practice some linked genes. In fruit flies, a gray body is dominant to a black body, and normal wings are dominant to vestigial wings. And the genes for the two traits are linked. Heterozygous flies were crossed with black and vestigial flies and produced the values below. And the question is, how many map units apart are the genes? So first let's talk about what if these were not linked genes? What if these were not on the same chromosome? What would be our Mendelian genetic probability for getting a fly with black body and vestigial wings? So our heterozygous flies are going to be, for body B and for wings W, have one capital and one lowercase. And let's find the probability of the body and the wings, and then we'll multiply them to get our combined event probability. So for body, we have capital B, lowercase b, and then they were crossed, of course, with our homozygous recessive black and vestigial fly. There we go. So then it's going to be 50% heterozygous and 50% homozygous recessive. And if we we do our cross for our wings, then we should get the same thing of 50% and 50%. So if, let's say, we were trying to find that our percentage of black and vestigial flies, so it'd be 1/2 chance of getting a black body and 1/2 chance of getting vestigial wings, multiply that out, and that means there's a 1/4 chance, or a 25% chance of getting a black and vestigial fly. Now, if we take a look, because these are both the recessive traits, now if we take a look at our actual numbers, you can see that gray normal had 778, black and vestigial had 785 flies, black normal had 158 flies, and gray vestigial had 162 flies. So as you can see, it is definitely not 25% that were black and vestigial flies from our heterozygous fly that was crossed with black and vestigial fly. It's definitely a lot more. So let's explain this using linked genes. And if we want to find how many map units apart are the genes, then we need to find our recombination frequency, which is going to be the equivalent 1% recombination frequency, which is the chance that there's going to be a recombinant produced, meaning a chance of crossing over, is going to equal 1 map unit, so how far apart are the genes on the chromosome. And we know because these genes are linked, the majority of the offspring are going to resemble one of the parents. They're either going to resemble the gray normal parent or the black vestigial parent, and these are going to be our parental types, while the offspring that don't are going to be our recombinants. So then if we are looking for our recombination frequency, I'll call it RF here, that's the recombinants divided by total offspring, and then multiplied by 100 to make a percent. And so the number of recombinants here would be adding up these two groups here, and that's going to make 320. And our total number of fly offspring is going to be 1,883, and that's going to come out to about 0.17, which is 17%. So the chance that we're going to get an offspring that is not a parental type is going to be 17%, and the genes are 17 map units apart. Now, time to talk non-nuclear inheritance and mitochondrial DNA. And non-nuclear inheritance is just like what it sounds, inheritance that is not based on genetic information from the nucleus, such as our chromosomes. And this is because mitochondria and chloroplasts contain their own genetic information, presumably from when they were separate organisms not part of the eukaryotic cell. And mitochondrial DNA is not passed down through meiosis. Mitochondrial DNA happens to be passed down because within the egg cell that is then fertilized by a sperm cell, there happens to be mitochondria in order for that cell to survive. So that egg cell mitochondrial DNA is going to be passed down to the offspring. So the only mitochondrial DNA in the zygote comes from the egg cell, and so this mitochondrial DNA only travels down the maternal line, meaning even though it mutates over time, you can have a rough estimate of tracing back all of the women on the maternal side of your family through the use of mitochondrial DNA, because it doesn't change except for mutation. It does not undergo genetic variation like genetic information within meiosis. Now, sex-linked genes. Now, these are not linked genes, but these are genes located on a sex chromosome. And in humans, that's either going to be an X or a Y chromosome. And most are born with either an XX chromosome or an XY chromosome, although there are cases where a chromosome is missing or there is an extra chromosome. And sex-linked genes are either linked to the X chromosome or to the Y chromosome, although more commonly to the X chromosome. And the X chromosome traits can be recessive or dominant in daughters because daughters are going to have two X chromosomes. So if let's say this is a recessive disorder, then if the daughter is heterozygous, then the daughter will not will not be affected by the disorder because there is another X chromosome which can be dominant, which will balance out or express dominance over the recessive trait. But it will always be represented in sons, because in sons there's only one X chromosome, so whatever trait that X chromosome has, then that's the one that the son will express. And that X chromosome has to come from the mother because the father is giving up his Y chromosome. And so fathers will pass X-linked genes to their daughters, but not to their sons. They will only pass a Y chromosome to their sons. And sons will express the allele on the X chromosome inherited from the mother. So over here we have an example. This is a recessive sex-linked or X-linked trait known as hemophilia, and we'll talk about some different traits in 5.6. But we have an unaffected father, and because it's recessive and he has a dominant, he's got the dominant X-linked chromosome, then he's going to be unaffected and he has the Y chromosome. Whereas the mother is a carrier, meaning that she has one dominant and one recessive X-linked chromosome. And so we can see here that this daughter is going to be unaffected because it inherited two dominant X chromosomes. This son will also be unaffected because he inherited the X chromosome from the mother that was unaffected and the Y chromosome from his father. However, this daughter will be a carrier because she inherited the X chromosome from the mother, which was the recessive one, the affected one, and this son will be affected because there's only one X chromosome slot, and it's the one that came from the mother because the other chromosome came from the father, and that was a Y chromosome. Lastly, we got to talk about pedigrees. And a pedigree is a diagram that shows the relationship between parents and offspring across two or more generations. And it can demonstrate which of the people within the family are going to have or express a certain trait and what gender they are. So as you can see here, we have circles representing females and squares represent males. And when the circle or square is shaded, that means that individual expresses the trait. And you can help predict the genome of future offspring. So over here we have this would be our first generation, then they had offspring, and then that generation had offspring. So we have 3 generations within this pedigree. And now we're going to answer this question. If individual 3,4, meaning this individual right here, married a man who was heterozygous for the trait, what percentage of their children would express the trait? So we know that this other person is going to be heterozygous, so we'll call the trait T, for instance. So they're going to have a capital T and a lowercase t. But in order to figure out the probability, we need to know what is the genome of individual 3,4. So let's do some math here. The original mother had the trait, however, the father did not. And so their offspring, they had 3 children, 2 of them had the trait, and the last one didn't. And now, in a family where both parents had the trait, then all of the offspring also have the trait. However, in a family where one offspring has the trait and the other offspring doesn't, then none of the offspring have the trait. And especially looking at this cluster right here, this can suggest that the trait is actually a recessive one, because if the trait were to be dominant, then there's a good chance that some of the offspring would be expressive of the trait. And so if the trait is recessive, that means any individual with a shaded circle would have to be homozygous recessive in their genome. So if we did that, for instance, lowercase t, then there's probably a good chance that this would be a heterozygous individual, capital T, lowercase t, and that way these 2 offspring would have the trait, while this third offspring, individual 2,3, would not be affected. So if we were to say that individual 3,4 has a trait, has a genome of lowercase t is homozygous recessive, then we do our Punnett square, capital T, lowercase t, lowercase t, lowercase t, and we see that 50% are going to be unaffected. They have the dominant allele, while 50% are going to be affected and would express the trait. So there you have it, non-Mendelian genetics. Next up, environmental effects on phenotype.